Okay, so welcome back to this next video in which we are discussing homologous recombination to fix our double-strand break. Okay, so we've now discussed the role of the mRN complex in signaling to increase the expression level of proteins involved in DNA repair. We now want to turn our attention to the role of the mRN complex in homologous recombination. Okay, so let's say we have our DNA here. Okay, so we've had this double-strand break occur. Okay, now it's going to be important in a moment that we understand the polarity of DNA. Okay, so in fact, let me draw the original piece of DNA again. Okay, so remember, DNA consists of these two strands, okay, and these two strands are anti-parallel to one another, so they run in anti-parallel directions. So let's say this first one here, the 5' end is up here, and the 3' end is down here, so the DNA is polarized in this direction, okay, and then the other strand will be polarized in the opposite direction, so this will be the 3' end, and this then will be the 5' end. Okay, so if we've now had this double-strand break occur, let's label up the poles of each of these fragments. So this side will be 3', this side will be 5', this side will be 3', this side will be 5', doing those first strands there. Okay, so let's colour these in blue here. And now doing the bottom ones, oops, this side is 5', this side is 3', this side is 5', and this side is 3' as well. So let's colour those in blue as well. That's going to become important in a moment. Okay, now, remember what has happened is our mRN complexes have bound to both of these free ends. Okay, so let's try and squeeze one of these in. In fact, actually, I will try and draw an mRN complex in here. So let's draw our mRN complex. So remember, here are these zinc hooks. Then we've got the coil domain coming down here. Then we've got our heads here. Okay, and then the coil domain comes here like so. Okay, and I'll just finish that off like there. So those are the Rad50 proteins here. So let's colour them in again. So we'll have the zinc hooks in orange here. Okay, and we'll have the heads down here in green. Okay, and then underneath the heads of the Rad50 proteins, we've got the two MRE proteins. Okay, and then underneath that, we've got the MBS1 protein. Okay, and I'll have that in yellow. Right, now, if homologous recombination is going to occur, what's going to happen is a certain protein is going to come and bind to MBS1. Okay, so I'll draw this protein here. And this is what's known as the CTLP protein. Okay, so we'll have this in vivid purple here. So this is CTLP. Okay, and remember this will be occurring on both sides. So this side will have an mRN complex as well, and it will be getting CTLP binding to it as well. Okay, so CTLP is essential to bind to MBS1 if you're now going to trigger homologous recombination. Okay, if you're going down the non-homologous end joining, that's a completely different pathway, and CTLP will not bind to the MBS1. Okay, right. Now, what's going to then happen is the mRN complex is going to start moving up the DNA. Okay, so this here is going to start going this way, and the one that then is around this side, which I'll just draw like so, that's going to move up like so. Okay, right, and it will move up by around 100 to 200 base pairs. Okay, so it's going to move right up the DNA. And then what it's going to do is it's going to make an endonuclease cut in the DNA. And it will cut the one which has its 5' end facing into the free end. Okay, so let me draw this picture again. Okay, so here we have our broken end of our DNA here. And here's our other broken end of the DNA. Okay, and remember the polarities. This is 3', this is 5', this is 5', this is 3 prime, this one's 3 prime, this one's 5 prime, this one's 5 prime, this one's 3 prime. Okay, now, the mRN complexes, which have now got CTLP bound to them, are moving up the DNA, and they're then going to make an endonuclease cut. And I probably should just explain what the difference between an endonuclease and an exonuclease are. Okay, right. So, let's begin with an exonuclease. An exonuclease is easier to explain. Okay, so, if we have a DNA strand here, and now this is just a single stranded piece of DNA, okay? An exonuclease is going to start from the end of the DNA, okay? So it's an enzyme, firstly, and it's going to start from one of the ends of the DNA, and it's going to cut off the first nucleotide of the DNA. Okay, so it's going to cut off that nucleotide, then it will move on to the next one, cut off that next one. Okay, so it cuts off nucleotides from the end of the DNA, basically. Now, you can have two different forms of exonuclease. You can have ones which start at the 3' end and work up to the 5' end. Those are called 3' to 5' exonucleases because they work from the 3' end and they go up to the 5' end. But you can also have 5' to 3' nucleases, which will start at this end here and then work their way down to the 3' end, and they'll be chopping off nucleotides as they go. Okay, so those are the two types of exonuclease, and we're going to need that definition in a moment. Okay, now an endonuclease is an enzyme which is just going to come into a piece of DNA here, or in fact it can be RNA as well, but we're dealing with DNA, so we'll keep this in terms of DNA. Okay, and it's going to come into the middle, and it's just going to cut one of the phosphodiester links in the middle of the strand, and therefore it's going to cut the piece of DNA into two separate portions. Okay, so that's the difference between an endonuclease and an exonuclease. Endo means inside, exo means from the outsides. So the exonucleases work from the outside inwards. The endonuclease just comes into the middle and then just cuts the strand in two, basically. Okay, so we're going to see that the mRN complex is actually capable of both endonuclease and exonuclease activity. Specifically, the proteins which are capable of nuclease activity are the MRE11 proteins. Okay, so what's going to happen is our mRN complex is going to move up the DNA, okay? 100 to 200 base pairs it's going to move up. And then what's going to happen is it's going to firstly make an endonuclease cut, okay? Now, it's going to change conformation so that the MRE11 proteins are brought closer to the DNA, because remember it's the MRE11 that's actually going to function as the nuclease enzymes. It's actually going to function as both an endo and an exonuclease. So we need it to be closer to the DNA. At the moment, the heads of the RAD50 proteins are the closest to the DNA. So what's actually going to happen is the mRN complexes are going to change conformation to something that will look more like so. Okay, so here are the zinc heads here. Here's a coiled coil domain. Okay, then we'll have the head of our RAD50 protein here. Okay, here's another head of our RAD50 protein. And now the two MRE proteins are going to sit in the middle there with the MBS protein underneath. Okay, so it's going to change conformation. Now, of course, I've missed something off here. We also need the CTLP protein. That's still going to be there. So I'll have CTLP here. Okay, right, so let's just colour everything in to try and show the parallels. So here is our head of the RAD50 protein. Here's the other head. Okay, so we've got our two RAD50 proteins. Both of them have heads. Okay, here are the two zinc hook domains of our two RAD50 proteins. Here are the MRE11 proteins in the middle now, and they'll now be in very close contact to the DNA, which will be coming in through here. Okay, And then underneath the MRE11 proteins here, you've then got your MBS1 protein here. So this is MBS1, okay? And then bound to MBS1, you then have your CTLP in purple here. Okay, right. So these two complexes are now one over here and one over here. Okay, they've moved up hundreds of base pairs away from the broken ends now. And what they're going to do, well, what the MRE11 protein is now going to do, is it's going to make an endonuclease cut. And it's going to make an endonuclease cut on only one of the strands. It's not going to do it on both strands. It's going to do it on one of the DNA strands. And the DNA strand which it will choose is the one that has its 5' end facing into the broken edge, basically. So if we look at the broken edge here, which I'll highlight in purple here, here is the 5' end here, and this one is a 3' end. So we are going to cut this strand here, the one whose 5' end is facing into the broken edge. Okay, similarly over here we need to look at which one has its 5' end facing into the broken edge. Well, it's this strand here, so we're going to cut there, basically. Okay, so that's the first thing that's going to happen. It's going to make this endonuclease cut. So that was an endonuclease cut. And now what's going to happen is the mRN complex is going to work its way back up. Okay, so it's going to make its way now back towards the broken edge. Okay, and as it goes, it is going to chop off this entire strand here. So it's firstly made this cut, but of course this fragment that it's made now is still attached onto the other strand by all the hydrogen bonds between the organic bases. Okay, what's now going to happen is it's going to work its way upwards here and chop off all of the nucleotides one by one. Okay, so it's now going to work through exonuclease activity. Okay, so it's going to work its way up, chop a nucleotide off, work its way along, chop a nucleotide off, work its way along, chop a nucleotide off, and it's going to continue that all the way along, basically. Okay, and the result of that, therefore, is that it's going to completely remove this section of DNA here, this single strand of DNA here, to leave this portion completely unpaired, so it's no longer going to have a complementary strand. Okay, and this is exonuclease activity. Specifically, we're working from the 3' end of this strand up to the 5' end, so it's 3' to 5' exonuclease activity. So MRE11, not only does it have the endonuclease activity originally, but it also has 3' to 5' exonuclease activity. And I hope you understand why it's 3' to 5'. We are starting at this end here, working our way up, chopping a nucleotide off one by one. Okay, so we're working from the 3' end and going up to the 5' end of this single-stranded piece of DNA here. Okay, and therefore this is 3' to 5' exonuclease activity. Okay, now, in addition, there are other exonuclease enzymes which are going to work their way in the opposite direction. They're going to come in at this point where the mRN has made this cut, and they're then going to work their way on this way, okay, going in the opposite direction. They are therefore 5' to 3' exonucleases, and they're going to work their way back and cut this strand up. Okay, now what is the overall result of this? Well, the overall result of this is that we're going to get these large overhangs, okay, like so. So let's work on our orientations here. Here's a 3', here's a 5', here's a 5', here's a 3'. So this strand, which has its 5' end facing into the broken edge, has been hugely trimmed back, okay, to produce this massive, great piece of single-stranded DNA here, okay, known as the 3' overhang. Okay, and this is generally around 1,000 base pairs long. Okay, and remember, that's why it's important to understand that not only does mRNA work its way this way, but also 5' to 3' exonucleases are working this way. A good example of one of those 5' to 3' exonucleases is exo1. Okay, so these other 5' to 3' exonucleases will work the other way to increase the size of the trim, basically. Because remember, I told you that the mRNA complexes only move up around 100 to 200 base pairs. So they're only going to trim off around 100 to 200 base pairs. Okay, so how do we create an overhang of around 1,000 base pairs in size? Well, it's because the other 5' to 3' exonucleases have trimmed off this other portion here. Okay, so let's colour in this strand in blue, and we'll have this strand in blue as well. Okay, now on the other strand, what's happened? Well, on the other strand, it's in this way. Okay, so this is the 3' end, so this hasn't been trimmed. This is the 5' end, and then the 5' end of this one that faces into the double strand break has been trimmed right down similarly. Okay, right, so we've created these 3' overhangs, which are massive then. Okay, how is this going to help in homologous recombination? Well, what is firstly going to happen is loads of proteins are going to bind over the single-stranded DNA strand here. Okay, so you're going to get a huge number of proteins binding all the way over this. So proteins instantly bind over DNA that is single-stranded, like so. Okay, and the protein that is binding here is a protein known as RPA. Okay, so I'll put this here. So this is RPA. This is the initial protein that's going to bind here, although we're going to replace the RPA in a moment. Now, RPA stands for replication protein A. Okay, so the R is for replication, the P is for protein, and then A is for A. Okay, so when you create these 3' overhangs, initially what's going to happen is you're going to get RPA proteins binding over the single-stranded DNA. However, RPA is not going to help with homologous recombination. So what we now want to happen is we want to take these RPA proteins off, and instead we want to put on a protein known as RAD51. Oh, and by the way, the mRNA complex is now detached. It's come off the other end, basically. It's worked its way along the DNA, chopping this bit off, and it's then detached off. Okay, what we're now going to do is we're going to replace these RPA proteins with a protein known as RAD51. Now, how does RAD51 get replaced on in place of RPA? Well, basically there is a protein which assists in the addition of RAD51 onto the single-stranded DNA. Okay, and this protein that's going to play a role in helping RAD51 onto the single-stranded DNA is known as BRCA2. Okay, which stands for breast cancer 2. So this is one of the ones that is associated with certain forms of breast cancer. So the BR is for breast, the CA is then for cancer. So this is breast cancer 2, BRCA2 protein. Okay, so basically the BRCA2 binds to the RAD51, and then it facilitates the exchange of the RPA protein off, and then the addition of the RAD51 protein on. And then gradually what you do is you replace all of these RPA proteins with RAD51 proteins. Okay, and this creates you something known as a RAD51 single-stranded DNA nucleofilament. Okay, so the name for a single-stranded piece of DNA which is covered in RAD51 proteins, just like this picture here. So imagine that these proteins are now no longer RPA proteins, but instead are RAD51 proteins. The name for this structure is to call it a RAD51 single-stranded DNA nucleofilament. Okay, right. And this RAD51 single-stranded DNA nucleofilament is now going to take part in homologous recombination. Okay, so now comes the killer moment. What are we going to do? How are we actually going to fix this double-stranded break in the DNA? Well, there is two different scenarios for how we're going to do this. One is better than the other. Okay, so we have lost some information here. There are potentially nucleotides that have been completely lost from the middle. Okay, this 3' overhang is not complementary to this 3' overhang, so do not think that we're now just going to stitch them back together. Okay, so where are we going to get this information from? Well, the ideal scenario, which is often true, which is sometimes true at least, is that the cell is currently in the later stages of the cell cycle. Okay, and if the cell is currently in the process of dividing, then as soon as the double-strand break has occurred, the p53 will stop the process of division. But the ideal thing is that if we are in this late stage of the cell cycle, we will have a sister chromatid made. Okay, so every single one of the chromosomes will have been copied if it was in the later stages of the cell cycle. So we would have two sister chromatids which would be absolutely identical. So every single one of the 46 chromosomes would have a sister chromatid. Okay, so these two chromosomes will be sister chromatids. Okay, so if one of them has then suffered a double-strand break, what we can go and do is look at the information on the other and use it to repair this other one. Okay, so if we do have a sister chromatid to the chromosome which has got this double-strand break in, we will do homologous recombination with that sister chromatid. What if we don't have a sister chromatid? Okay, so if we don't have an exact copy of the chromosome, what can we do? What is the second best thing we can do? Well, although we don't have an exact copy, we have the other homologous chromosome. So remember, in the nucleus of your cell, you do have 46 chromosomes, but really it's 23 pairs of chromosomes. So you have two chromosome 1s, two chromosome 2s, so these are chromosome 1s, these are chromosome 2s, all the way down to chromosome 22, and then it's a little bit more complicated for the sex chromosomes, so we won't go there. Okay, but the point is, certainly for the autosomes and potentially for the sex chromosomes, I mean, if you're a female, you do have two X chromosomes. Okay, if you're male, then you'll have an X and a Y, which have totally different genes on, so it doesn't work as well there. But for all the autosomes, which are the 22 non-sex chromosomes, you have two homologous chromosomes. Okay, so these are the homologous chromosomes. Now, the homologous chromosomes are not identical, absolutely not. They will have different sequences of organic bases, okay, without a question. However, they have the same genes on them, okay, and certain portions of them will be very, very similar, if not identical. So they will have a lot of similarities between them, okay? So we could hope that this region that has been broken here has effectively a homologous region in the other chromosome, okay? And that's our best hope for trying to get some restoration of this information. Okay, you might think, well, couldn't it go to the neighbouring cell and use its genome? Well, no is the answer to that. But instead it's going to go to its own homologous chromosome and try to rebuild this damaged area from that homologous chromosome. Okay, so if there isn't a sister chromatid, we use the homologous chromosome with the hope that there is a region in the homologous chromosome which has a similar enough sequence of organic bases, if not identical. And it most likely will be identical, but there will be differences between the two homologous chromosomes. Okay, but the two homologous chromosomes have more similarities than differences. Okay, so what then is going to happen? Well, let's draw this picture then. So we will take this strand here, Okay, this portion here with this 3' overhang, we'll leave this one out for the moment, but it can do the same thing. But actually we only need one to undergo the strand invasion process, so we'll just say it's this one. Okay, so we have got this chromosome that we are going to use to reconstruct our broken area. Okay, so let's colour this in. So we'll colour in the two different strands now in different colours. This is going to help us in a moment. Okay, so we've got one strand in orange and one strand in turquoise. And let's say this region of this chromosome, whether it's a sister chromatid or a homologous chromosome, is the corresponding region for this portion that has been broken here. Okay, we then have this massive great 3' overhang now that is covered in Rad51 proteins here. Okay, creating a Rad51 single-stranded DNA nuclear filament here. So let's colour this in blue here. Okay, and then we'll colour in those Rad proteins, I think, in green. So these are Rad51 proteins here in green. Okay, now, what's going to happen now is a process called strand invasion. Okay, and what's going to happen is the Rad51 protein is going to catalyze the opening of this strand, okay, and the binding of this 3' overhang to the portion that is hopefully going to be identically complementary to this 3' overhang here. So remember, this is the 5' end of this strand, this is the 3' end, this is the 5', this is the 3' end. So a portion on this orange strand here is going to be perfectly, we hope, complementary to this portion. It's certainly true if it's a sister chromatid. It will hopefully be also true if it's a homologous chromosome. Okay, and what we're going to do is we're going to break the bond between this orange portion here and this turquoise strand, and we're going to say, okay, we want this orange strand now to bind to this blue strand, this 3' overhang of this blue strand, rather than the turquoise strand here. Okay, and this process is known as strand invasion then. Okay, so let's draw this happening now. So as we get the strand to, as we get this 3' overhang to bind to the orange strand, the Rad51 proteins will gradually dissemble off, okay? So the Rad51 proteins have now gone, they've done their job. Their job was getting the orange strand to do what it's doing now. Okay, so it's now come away from the turquoise strand temporarily. Okay, and instead it is bound to this 3' overhang of this blue strand here. Okay, like so. So here is the orange strand once again. Okay, and here is the turquoise strand here. Right, okay, so let me just get this straight. Okay, so now what's going to happen is we are going to use this orange strand to extend our blue strand. Okay, so remember the problem was we don't know if things have been lost from here. Okay, so we are now going to replace whatever information we have lost there by extending this blue strand to be complementary to the orange strand. And by the way, this new loop that you've created here, this is known as a D loop. Okay, so this loop here, where we've got the blue strand infiltrating in this way, this is called a D loop. Okay, so what then is going to happen is we are going to extend this blue strand. Okay, so DNA polymerases are going to extend this blue strand on to build a complementary strand to this orange portion here. Okay, and this can happen because this is the 3' end here. Remember, DNA extension always occurs by adding the 5' end onto the 3' end. Okay, so what we're now going to do is extend this blue strand further on. Okay, so you're going to get strand extension. Now, as you get strand extension, the strand towards the end is going to detach from the orange strand. So let me firstly just show strand extension, and then I'll talk about the separation further back. Okay, so here we are now synthesizing more blue strand, which is complementary to our orange strand here. Okay, so let's colour this in again. So in blue here we are extending this strand further onwards. okay, and building it so that it's complementary to our orange strand here. Okay, and as we do, we will be peeling the orange strand off the turquoise strand more and more. Now, as we do this, and this is the process of strand extension, which will be catalyzed by DNA polymerase enzymes, as we do this, we will also gradually be we'll be peeling this blue strand here away from the orange strand at the back here, and then we'll be reattaching the orange strand back to the turquoise strand. Okay, so we'll continue the extension on this end, and we'll continue reforming our normal chromosome back here. Okay, so what will start to happen is this. We'll start to get the orange strand just over here now. Okay, and now the 3' overhang here is no longer attached to the orange strand. The orange strand has gone back to being attached to the turquoise strand. Okay, right. So we are now extending our blue strand by using the genetic information within this other chromosome, basically. Then what you're going to do is you're going to detach the blue strand away from the other chromosome. Okay, so you're going to bring this longer piece back now. Okay, so now we've extended our 3' extension hugely here. Okay, and we haven't damaged the other chromosome. The other chromosome has now been formed back again. Okay, and the hope now is that what we've done is we've extended this 3' portion so that now it has a portion which is complementary to this 3' overhang here. Okay, so the two broken ends can now re-anneal together, and then all we need to do is use DNA polymerase to extend this portion up, so to fix this in. Okay, so it will fill up that gap now, and then it can also fill in this gap, because remember it's adding always nucleotides from their 5' end onto a 3' end like this. So it can extend this region like here, synthesize right to there, and then of course DNA ligase will fill in the final gaps there. And that then is how we can repair a double strand break like so. And this gets around the loss of the genetic information that we have potentially suffered there, okay, because we use the information on the other strand to reproduce this lost piece of genetic information in the middle, okay, and this will of course always work if we've got a sister chromatid, and it will generally work if we use the homologous chromosome as well. And then what we can do is stitch the two sides back together, and this, unlike non-homologous end joining, really can reproduce the exact chromosome back again, okay, just good as new basically. Okay, so it's better than non-homologous end joining, but non-homologous end joining is more commonly used.